Okay, we are still waiting for the catching up participants to reach. So I'm going to consume 5 minutes to wait for the colleagues, your colleagues. As I'm meaning myself and Professor Saw, organizers. And also, also in charge of the taking care of 11 students who enrolled half of our projects related with brain. And then one student, they registered the nuclear medicine graduate course. So we consist of really the heterogeneous group or origins and expertise. So in that sense, the nuclear medicine is a most common commonality. Why? Because finally, we are going to be with 30 connection sites and 30 plus alpha, meaning the less than 40 participants. And now we know that these are the participants once ever before until now, 217. And YouTube is going to be another media for us to communicate with you. So YouTube is going to be a kind of platform type of webinar, which is used by the European Association of Nuclear Medicine or Society of Nuclear Medicine and Molecular Imaging, both of the huge societies with very novel information. They do open webinar type of the platform for the continuing medical education for even for us, meaning the Asian population. So it was a shame or pity to have a webinar type. Why? Because its webinar type is kind of there are brilliant people who are talking with each other. Sometimes we are chosen to become one of them as a kind of a best abstract submitter from this country, and the primary problem is the time zone difference, and the secondary problem is the webinar type of format. So I need to just overcome that kind of shortcoming in this interactive, the desirably interactive one. So I'm going to use my two occasions this week, today until 2:50. So I'm going to use my entire time without break. It was supposed to be 2 hours and a half, but it was supposed to be 2 hours and 3 hours. In that case, the 50 minutes and 10-minute break, and 50 minutes and 10-minute break, and another 50 minutes and 10-minute break. I'm going to use 3 hour course. So meaning the 2:50, we are going to close. So if you reach 2:50, then the 무슨 일이 있어도, meaning we are going to abruptly stop there. Why? Because I have another opportunity next week. So today I'm going to experiment the upgraded version of this one. If any question or any kind of comment pop up in your mind while I was talking, you can just write down on the chat window. Then we are going to read and repeat your question, and so that everyone can be reminded, and then we are going to discuss about that. However, if this is going to be a little bit more interactive, good morning everyone, and especially only for Korean good afternoon. So why? Because it's just 7 minutes after noon. So the good morning, and then you're going to invest precious time, the morning, the hours for practice and training yourself while you're in a resident position or the junior staff and faculty, and in whatever the discipline or whichever institution you are in. So I'm going to just kind of experiment another format. That's the reason why I just sent you the void only with the titles, void the text, actually PowerPoint to each group of you. Dr. Gil, for one single person, and 11 students in participating here as their graded course seminar or the workshop or the lecture. And then the 30 or more institutions as the next generation leaders of nuclear medicine in Asia from the possibly 6 or more countries. Anyway, we are waiting for TAIN report. We have been chosen as a short in the short list, so we submitted. Meaning the Professor Hall composed the entire project and to be submitted to TAIN, Asia Connect, and then it is now being as I know, evaluated by the European colleagues, so that they need to decide whether this project is going to be further supported, so that the new coming countries, new coming institutions are going to be supplied the video or conferencing equipment, and a minimum one, and networks are going to be used. All right, so this might be a new information for you, and then at first I'm going to just repeat mine, and then with this kind of experiment, I'm going to just enhance the advantage of participation. What I'm saying is just even though there is a possibility or opportunity to look over or review the YouTube or the PDF or PowerPoint slides on the homepage later on, but on air participation should have some advantages, so that your voices are to be heard by the lecturers. Since Professor Edmund Kim's lecture last year, this is the 3rd semester, and then now the number of participants are stabilized. However, those almost 40 sites, 40 connections, beside another 10, the graded course students, and for students, it is a responsibility for them to participate, but our Asian colleagues, they don't need to participate, and they do allocate or save their time after working hours, and then to listen to the YouTube one. So if it is going to be webinar, and there is no merit, And so interactive, the one is the next step. So I think then I'm going to just rehearse it. How I already asked Dr. Gill to become the fixed discussant in several medical problems, and I just asked my graduate students to answer to my questions. If I'm going to deal with the science issues, then just once per occasion, meaning today once and next week once, and then their privilege or their chance to participate and express their understanding during this course is going to be up. However, what I'm going to really need is just to encourage you to get involved. So in that sense that I'm going to just ask anyone whose name is quite familiar to me, like, is there Dr. Karishima from Indonesia? Why? Because I am seeing her name very often, almost every day, during BBC television, the Asian business section, and Ms. Karishima is running as an anchor of that program, and I'm seeing her the face every day. And I remember her name, or Andreas Lim, or Alexander. So you can just ask, or you're going to be asked by me. All right, please don't be threatened. This is not a kind of enforcing anything to you, but just kind of please help me. So let me start the science session, the lecture part, and the first one is, as I just told you, all those contents which were dealt with by Professor Yu-Kyung Kim, she, and also Dr. Son, she, and Professor Kim, he, those experts, the best experts in our country, not world, but their level of expertise in some parts already reached the top level. And so they could just kind of summarize their understanding to you. And last week, you have already listened to a junior professor of this department, meaning Seoul National University, but however, in different hospital, Bundang Hospital, Professor Song. So I assume that you have listened to 5 lectures, but did not understand anything, did not remember anything. So I just reviewed that one. So from the standpoint of the problem formulation, why they are explaining their expertise regarding some issue. And those issues, many of the issues, in many of the issues, you would be already familiar. So you can just kind of consolidate your knowledge. However, in some cases, in some points, in some issues, in some items, you might have heard novel information and how to integrate those into your own problems to be solved, not right now. Currently, your institution falls short of any instrument or any kind of a facility or any kind of platform, any kind of test bed to try, but sooner or later, or not at all, finally in your country or in your institution, you're going to solve that problem. So why not? The Asan Medical Center is larger than the Seoul National University Hospital and more modern than ours. So their level is almost reaching not Harvard, their level of the luxury, which is the level of Raffles Hospital or the Dubai or the Stanford. No, I don't think so. Stanford science is strong, but the medicine is not. Why? Because they are fewer people than the MD Anderson Cancer Center or Sloan Memorial Sloan Kettering Cancer Institution. Harvard, the MGH Hospital is very old, but their science and their expertise in medicine is very high. I had been to a small hospital in the Midwest of United States 28 years ago now, and I was really impressed. And I just kind of determined. I actually, based on that kind of impression, I came to be determined to make our hospital to be better than theirs. Why? Because I need to take care of my own people. So that's a problem formulation, and problem is going to be dissected, and then contexting the problems to the knowledge of in the literature, and that the third parts had been done by Professor Kim, Son, and Song. But as I told you, that kind of information would not have been integrated in your current knowledge. So I am going to try to quench the problem. So quench, the verb is used for quench the thirst. If you feel thirsty with many many kinds of lack of understanding of what you are hearing from the South Korean experts, so then please let us know. So that the Professor Seo is going to solely organize the next semester's one based on your wish lists. And we are not going to continue this kind of webinar type. So we can just shrink the 30 connection site into 10. And then we are going to exercise the interactive one. So how? Raising questions and inviting answers. Let me try it. Please don't be shy. You know, I have used this kind of information to the blank our third-year graduate students for 30 years. And some of them remember the strategic, lecture, kind of practice sessions in nuclear medicine department as kind of question and answers. Some of them told me that they felt bad. Why? Because they could not answer any of my questions. Why? Because none of my questions are listed in the textbook. If those are in the textbook, there is no need to discuss in the kind of situation, meaning the bedside, or in our case, in front of the computer monitor. Okay, so let me continue my topics and issues. I have brought this one from the nmportal.org. 10-2021, the May 26 brain theranostics update session, I have tried to reach here, but I could not say anything about the topography of connectomics. I could not say many things about the abnormal protein imaging, and those abnormal protein imaging was dealt with by Professor Yu-Kyung Kim, and undesired flaws thereof. So there was a real points. And the May 26 is the before American, meaning US FDA, decide aducanumab, whether aducanumab is going to be approved or not. So that approval was done, as is well known, the June the 6th. So I'm going to start from that point. And new targets and spatial transcriptomics, that's the kind of a little bit sensational and brand new topic. So I just to raise that in the top, the title, but don't be frightened. Don't be just overwhelmed. Why? Because spatial transcriptomics is not ours. Spatial is ours. Space, image, that's ours. And new targets is a kind of a holy grail. We need to find that, in everyday routine, thyroid scan was a popular and it's gone. Before thyroid scan, around thyroid scan years, and the liver scan was popular and gone with the introduction of the ultrasonography of the liver and thyroid too. And then, the bone scan is still there. And myocardial SPECT has been risen, and only in United States, only in US, they are doing 10 million cases annually, not by nuclear medicine physicians, but by nuclear cardiologists. They call themselves nuclear cardiologists. Actually, they are cardiologists. They have been interventional cardiologists, or the echocardiographers turned into nuclear cardiologists. Why? Because they had been so huge demand in US. So the nuclear medicine is practiced in US by a cardiologist half, and the other half is done by nuclear medicine physicians. And nuclear medicine physicians recently tried to find a way of breakthrough to keep their job alive by just adopting European the pioneers' work called theranostics. However, theranostics had been applied to many disciplines, including everything. So liver and thyroid and bone and many kinds of cancers, and also heart, as well as brain. But interestingly, the brain was a kind of a prodigy 30 years ago. The first, the science paper was published by the late the Henry Wagner of Johns Hopkins University, 1984, the first human receptor imaging by methylspiperon, C11 methylspiperon. At that time, it was kind of really novel. But after 30 years, they found that the brain application of nuclear medicine is scanty. So nobody is saying that I am brain experts, and also I'm the practicing clinical works regarding brain. Really? No. So new targets in brain should be understood based upon the common objective by the world society, world community of nuclear medicine, to make brain area to be prosperous for their clinical the application. So new targets and spatial transcriptomics, don't be the overwhelmed, don't be just alienated. Why? Because I'm going to explain it as I'm the novice and first mover to just adopt and understand using my last 6 months of full time. Owing to the COVID-19 lockdown, I could save a lot of time to research and to solve this problem, the brain theranostics, current and promises. And I'm doing the promises, and I have a privilege and responsibility to let you be exposed to this one. By just kind of lamenting. So I'm going to review the questions. And the first one is, which target of Alzheimer's disease for brain theranostics? Is it amyloid and tau, really? And issues of debate and dispute regarding FDA's approval of aducanumab, the monoclonal antibody, canumab meaning the human antibody, meaning in humans, autoimmune antibody had been rarely found, and the hypervariable portion of the autoimmune antibody, not popularly, but ever found to exist in human, was used as a kind of epitope for targeting. So aducanumab issue. And then we need to just understand Alzheimer's disease is not the only the cause of the dementia, neurodegenerative dementia in the elderly. And then the first competitor to Alzheimer's disease is not the Parkinson dementia, is not the rare diseases, but a little bit of common diseases of frontotemporal dementia. Frontotemporal dementia, the Maurice Ravel from France. Maurice Ravel had suffered from frontotemporal dementia in his late years. Maurice Ravel is very famous, the French composer who composed Bolero. So I think everyone would know Bolero. So that's kind of a repetitism is one of the symptoms, and so Bolero has been composed late in his years. So some neuroscientists, neurologists, just kind of as a diversion, they have written down that Bolero had become an easier version of previous the complicated repetitism of Maurice Ravel. To become much easier. Why? Because he get more sicker, more ill, and thus he could not use so many kind of refinement of those the repetitism. So everyone could understand. That's the reason why Bolero is using repeat and repeat and repeat and repeat and eternally. And everyone is hooked up by that melody, and everyone know. So we need to just understand the region or peptide specificity of images. And then off-target effect. Off-target effect, I have asked Professor Yun-Sang Lee last summer, after listening to his explanation, his kind and easy explanation of interpretation of chemistry. Then I asked him, and he could not answer immediately. How they would have found, how the scientists would have found that some of the imaging agents are targeting not the amyloid or tau, but the monoamine oxidase B. So what's the similarity between the plaque or the tangle with that one? That was the question one, and maybe I need to use almost the first hour, meaning first 1 hour, to explain question one, and then let's move on to the next. The second question was, which alternative target in AD for brain theranostics? So it is going to be, so everybody is saying that there is the cells, glial cells, which might have been involved and which had been ignored. So they say that PK11164, meaning C11 labeled the TSPO. Yeah, the TSPO is just a kind of a mitochondrial outer membrane, the peptide, and which is targeted by several the well-developed chemicals, and that chemicals were radio labeled, and we call it them radiochemical or radiopharmaceuticals, and some of them are in clinical use. But is it really the microglial the target? I mean the ligand. And what about the astrocytes? There are 4 brain cell types, each occupying 1/5 of the population. One is neuron, the other 1/5 is occupied by astrocytes. Another 1/5 is occupied by microglia, and another 1/5, much more localized, is by oligodendrocytes and its precursors. So oligo and dendrocytes are always accompanied by the OPC, oligodendrocyte progenitor cells. Why? Because oligodendrocytes are the proliferating the activity. So it should be supplied. Why? Because it's a kind of a handy person type of why the oligodendrocytes are taking care of the integrity of neurons. Then astrocytes and microglia, are they? Yes, 2. Then I'm going to say a little bit about TSPO, and then blame the scientific community to make our attention to be mainly focused on only TSPO and not other targets. They failed. You know, in the history, NIH has produced, has just kind of presented and has just supplied, no. NIH has given birth of the world-renowned the radiochemist, whose name was kind of demigod. When I just started the nuclear medicine 31 years ago, finishing my PhD, I started the nuclear medicine in 1990. At that time, NIH, the best radiochemist, the pioneers, previously pioneer, now the great leader was Eckelmann. That information was given my colleague, Professor Jae-min Jung, who once was the president of the world. They do not use the word. President of the, it's world organization, president of the Society of Radiopharmaceutical Sciences. It's SRS. Professor Jaemin Jung. At that time, he was a PhD student, and he told me that there is a Greek guy named Eckelmann. His major is muscarinic receptor imaging. Why? Because the dementia patients are suffering from the problem in cholinergic the communication. Cholinergic communication was the best one. Still in the dementia field, only the drug, sole drug, which had been approved by US FDA and European Medicine Agency, EMA, is Aricept. Aricept. The Aricept is being used. It's a kind of really long-standing blockbuster. Drug blockbuster, maybe tens of $1 billion per year. The dementia patients cannot help the taking that pills. Why? Because there is no other treatment, including my mother-in-law. She was diagnosed as the mild cognitive impairment 5 years ago. She's now suffering from the downhill, still taking care of the everyday living, but she asks the same questions during meeting with me or encountering with her family during 30 minutes, the same questions 10 times. We answer to her questions, we meaning I and my wife, and her grandson's granddaughter are willing to answer to her questions 10 times as if we listen to the question for the first time. Okay, so she's taking the Aricept, and we don't have any remedy. So this kind of situation should be improved. Not in my lifetime, but in your lifetime. You belong to the next generation of the nuclear medicine. I'm not quite sure how long your country's, your people's longevity, the life expectancies, how much they have reached currently, meaning in Korea, it's higher than the 80 years old. So the baby now is born, and then his or her life expectancy is 80 years or more. So it is going to be your present, meaning your situation, and then in your family, like me, somebody would luckily the other three, the parents of me and are well. So the only one per four, but we are expecting the prospects for prognosis as time goes by. And then after the 4 groups of brain cells, then what about the other cells? Meaning are there any immunocytes? I call it immunocytes. I'm not calling it myeloid cells or lymphoid cells. So immunology has been expanded almost marvelously during the last 3 decades, meaning the understanding of the immune cells, including microglia, has been totally changed since my the medical school years it's been totally changed, and the total change has been has taken place, during the last 5 years, during the last 10 years, or at least just during the last 20 years. That's the year. In 2000, after completing my task, which was given my the senior, my mentor, the junior mentor, my senior mentor was the Professor Chang-Sun Ko. So that's the reason why we have Ko Chang-Sun Fellowship. And then the mentor, who's junior to Professor Chang-Sun Ko, late Chang-Sun Ko, is Professor Myung-Chul Lee. He led me to do the microspect and heart studies for the first 10 years. So in 2000, I have changed career to proceed brain. Why? Because brain is a blue ocean, still very much blue ocean. That is the problem. During the last 20 years, oncology, nuclear oncology, or the tumor stuffs, and even recently, since the pioneering work of the professor, now retired, Richard Baum. So that's kind of a German pronunciation, not Richard, Richard. Richard Strauss and Richard Baum and Johann Sebastian Bach. So that's kind of his real name. And since 20 years ago, theranostic had become a reality. But the brain theranostics, anything of them had become reality. No. So I almost spent 30 minutes or more so that everyone can just listen to the real contents of my first hour presentation. So the question number 4 was the real theranostics. And then around that time, I found that you don't have any factual knowledge. So even though the medical education should consist of the skill or kind of a decision knowledge, however, without factual knowledge, which is even especially novel, it is going to be kind of the speculation, and it is going to be a kind of too much risky, dangerous one without good factual, the good understanding of facts, not big lie, not fake facts. Fake facts are produced famously by Facebook, and then the U.S. Congress House representatives had recently decided to interrogate Facebook and Zuckerberg. So I'm expecting the better situation, though I'm not doing any Twitter, any Facebook. I belong to the older age. So we need to check the facts. They call it fact check. So I just turned into listing fact check instead of the problem formulation, so that we can just kind of help. I can help you the basic facts, help you to understand the basic facts with much ease, meaning you don't need to read Nature, Science, Cell, or any kind of neuroscience journal, but you understand the current progress because of me. Then I would be very happy to be a help to you. So please allow me to please accept my just kind of humble wish and humble trial. Okay, so I just summarized what I'm going to do today and then next week. And I have prepared, as is shown here, 56 slides containing facts, containing the dissection of the problems. And if you don't understand that, then please let me know. And at first, just not to embarrass you, I'm going to correct one issue here. This one, to our the resident student, Dr. Gill. In his the kind of term examination answer sheet, he answered based on the answer, the ependymal cell has a gap junction, and also I would have remembered a little bit incorrectly, but please excuse me. And because of this typographical error, this is a choroid plexus here from the lateral ventricle, third ventricle, and fourth ventricle of the brain, either a mouse or a human. Here they have mistyped. This epithelium has an epithelial cells, not endothelium. And blood vessel has an endothelium, yeah, with gap junction. That epithelium has a tight junction with proper claudin. Claudin and occludin is a protein which is just kind of zipping the cells and cells side by side. That's the reason why larger molecules cannot easily penetrate paracellular space. So let me just correct this. Dr. Gill, now you know?
Yeah.
Yeah. So you have you did not you did not do anything wrong. That's kind of a typographical error. I did not comment. So there was a 2019 neuroscientist, a Japanese author. I don't think the Japanese author would have made no, maybe because this is a figure and it's kind of original mistake. The first question, the continuing this one, I had shown my kind of favor and goodwill to Dr. Gill. So let me just ask you, this is a kind of which target for brain theranostics. So based on your current understanding or current information gathered from everything and internet or the mass media and the CNN and BBC and the Korean television news or newspaper or the textbook and whatever. So amyloid, so the which target do you expect here? Dr. Selkoe is one of the great guys whose name is a little bit weird, so that we can just kind of memorize it easy. Selkoe. And treatments for Alzheimer's disease emerge. So I think he would have been happy with the approval. He belongs to the major, the group of the community. So Dr. Gill, which target do you expect the next generation promising for AD therapy?
Maybe tau instead of amyloid beta.
So 1/3 of the community is saying, and among them, big shots, and leaders and frontiers are writing a review article that the tau is the next target. I am against it. Pursuing tau is going to waste another 20 years. When you are going to when you are coming to the age, when you are going to be promoted to honorary professor or retirement, you'd find yourself the tau pursuing has wasted all of your life. So you'd be frustrated like me. I hoped 20 years ago that during my lifetime, there might be some the treatment or disease-modifying drugs for Alzheimer's disease. So there are many problems. Amyloid plaque is the culprit, A-beta or plaque. So according to the Dr. Gill's answer, amyloid plaque is gone. Then why? Why treatment for Alzheimer's disease, aducanumab, has been approved? So we need to understand that situation. Then you just said tau. Is it going to be tangles, intracellular and extracellular? And you can just see the intracellular one. However, there are maybe Dr. Selkoe is a best expert in amyloid, A-beta oligomer, but he just opened the way to microglial cells or astrocytes here, very small. Is it really small? In his mind, neurons are big. We could find, and amyloid plaques are there. Sometimes it's called neuritic plaque associated with the extracellular tau. So recently, interestingly, intracellular tangle is a benign, innocent, just a waste. So without the intracellular tangles, the same pathology and behavior abnormality can be derived from a mouse model. So tau tangle, which one is abnormal? And then tau, we know that the mutant, there are many mutant tau. There are 3R or 4R repeat domain, and many prolines. Just ahead of proline, there are serine and threonine, and those serine and threonine has a hydroxyl groups. Those hydroxyl groups are phosphorylated and hypophosphorylated tau, and there are so many the prolines, and before proline there are so many serine and threonine. So we have so many modification of P, and in addition to phosphorylation, we have so many post-translational modification. They call it PTM. Even glycation and glycosylation and arginylation. Wow. So people have pursued. Professor Edmund Kim recently commented in our morning conference that that kind of complexity is a kind of 누가 좀 가르쳐 줄래요? 축복이 영어로 뭡니까? 갑자기 축복이 안 떠오르네. I just asked the English translation of some word in Korean.
Blessing.
Yeah?
Blessing.
Yeah, blessing. So Professor Edmund Kim told the complexity, some problem is a blessing to the scientist, but a kind of a menace, omen for clinicians. So thank you, Professor Cheuri. And then what about the spread? Eastern Coast, the neuropathologist called Virginia Lee, who's originally from the Taiwan. She's one of the best experts in the neurodegenerative disease pathology. And she just popularized since 10 years ago, there is a peptide propagation model. And because of the two Nobel, the prize had been given to the prion disease. Interestingly, Professor Virginia Lee, and her husband is also neurologist, neuropathologist, and the neurologist Trojanowski, who is the kind of inventor of the ADNI, Alzheimer's Disease Neuroimaging Initiative. And both of them are true leading experts in the world. And she has made a proposal, at first Parkinson's disease, and then tauopathy. So she did not care amyloid. She just kind of, just like a prion disease, prion, Jacob Creutzfeldt, mad cow disease, that's a prion disease, and spread. And then what about the other glial and immune cells? In my lab, under the supervision, under the leadership of Professor Cherry, just last year, we found some monoclonal antibody had been effective, effective against AD in mouse model. That's immune cells. Then what about the glial cells? Many people are doing research regarding the role of glial cells, including 3 glial cells, 1 astrocyte, 2 microglia, and 3 the oligodendrocytes and oligodendrocyte precursor cells. Okay, then I'm going to move on to the next issue. And then explaining this one, I'm going to take a 10 minutes break. You know, I have used only 5 slides to tell you the formularize. So the next the second hour after this break, I'm going to ask you, and so that just kind of test the connectivity using the video conferencing system. So let me explain it this one briefly. There have been the editorial by Permuter. Permuter was in Washington University. You know, interestingly, I had been there 28 years ago and stayed there 1 year. Permuter would have been there as a junior, the neurologist, now senior. The title was the FDA's green light and science red light. And you know, the Cell-Coes article, in essence, A beta of whatever species effect rather than cause. That's the another best experts, the rebuttal to Cell-Coes article here. For many chronic disease, but confer modest clinical benefit, and one must walk before one can run. Meaning, Cell-Coes is supporting the FDA's approval. However, everybody is not agreeing with him, including me. It's lamentable. One must walk before one can run, and using that kind of the unpredictable drug to the Alzheimer's disease patients and family, giving them false hope. After 10 years, you are the worker, we are going to run. Sorry, you have lost a lot of money, and you have lost your family member's life and kind of a quality of life, and also the life itself, and confer modest clinical benefit. He is a dangerous person. He was a best scientist, but he is far from clinical. And because of his fame, Science has invited him to write something, and it's been it's been challenged by another neuropathologist in North Carolina. And this was the editorial by Perlmutter. Side effects of the proposed dose included localized brain swelling, which should be monitored closely by MRI, repeated MRI, in 35% of the clinical trial participants, even microhemorrhage in 20%. When all this was put to the vote by the advisory committee to US FDA, 10 among 11, 10 voted no, 1 voted uncertain. It just kind of give up. No one voted yes, but they decided to approve that. And according to the information along the internet, if this succeed by some luck, this might be a real breakthrough. However, if this fails after 9 years of post-market survey, then this might be the second huge scam in the history of the FDA. The first one was the oxycodone by Duke. Oxycodone was found to be fatal and increasing the case fatality due to morphine addiction. The combination of fentanyl and then oxycodone was the main issue. So there was a recent US scam. And then this might be the second scam. It is really, really saddening and also frustrating. As a member of this advisory committee who resigned in protest over the decision of grant approval, I am still trying to fathom how this happened. US FDA jumped the gun by granting accelerated approval this month to Biogen's pricey, questionably effective and possibly harmful new drug aducanumab, a decision supported by not one of the 11 members of the agency's expert advisory committee. That was the situation. This is the second reason I do not authentically trust US decision. The first one was the Trump. The second one was this decision. Otherwise, I don't have any kind of the occurrence. Okay, let's stop here. And unlike I have just explained, I just asked one question to one participant. But after this one, please help me to just. This is 1005, and let's come back 115. And I'm going to ask at first Asia, and then where is it? Then Alexander, and then Asman, and then I have seen the Karishima. Okay, but now and then that's kind of I'm not going to ask questions. I'm going to invite your question, right? So that question might be just like, why do I had raised this kind of question, meaning Q1 and Q2, Q3, Q4, Q5, why it is important? That might be a good question. So meaning that you are interested in the subject. You're not interested in question 1, 2, 3, and you might be interested in question 4, especially. So that might then I'm going to jump to Q4, and then there is no the order index. Okay, please come back the 115. Bringing your coffee or tea or or water. See you after 10 minutes. Okay, thank you for coming back. The problem of the Professor Selquest's opinion was may provide and may enable and must work before. May work before one can run. So because this was a kind of a to the leftist extreme or the rightist extreme. So the other part, another best expert is A beta of whatever species is effect rather than cause, meaning the removing A beta does not So have you ever known that this kind of dispute there? No? Then please be reminded. So I was trying to just kind of remind you this one, NIA, AA, the research framework, several years ago. Right? But please remember that this is a research framework, not clinical guideline. Clinical guideline is produced by the American Association of Neurologists, AAN, or other organizations. And Professor has just given this information. So, but this information has a very good understanding. They represents the amyloid PET, meaning the plaque PET, and also tau PET. What kind of tau is visualized? That's the problem that I have that raised in my the question that raised that rose in my mind when I just heard tau PET and FDG PET. Yeah, we know that. Really, at the end of this 2 week course, the last slide is just questioning FDG. Right? Okay. So amyloid is making plaque. Yes. Plaque is not pathogenic, according to this expert's idea. And I just put not significantly pathogenic. Then tau. Do we know tau? No. Tau consists of the many tubulin, microtubules, and neurofibrillar tangle here, or neuritic plaque tau. Neuritic plaque is extracellular tau. Oh, when I was in my medical school, I learned that tau is making intracellular inclusion body called neurofibrillar tangle, and there has been no saying that tau is a kind of a extracellularly secreted or not. So that's the reason why I just started to learn. The low and high molecular weight using sarcosyl soluble and insoluble, or the nucleation, elongation, and paired helical filament, PHF, that kind of structural ones, and it's kind of a structural biology or the biophysics, and also biochemistry stuffs, as I just told you. Non-Alzheimer's disease CSF, in human studied AD CSF, there has been a whole variety of a phosphorylated tau, and there are so many tau, phosphorylated tau antibodies in vitro Gen 1 and Bartholomew, and our student had said that he has this one, and that kind of issues, and tau is secreted finally 2019 in Tau Biology book. This is a book and textbook, a monograph, and tau secretion is being done, the transsynaptic spread. So some scientists are favor this kind of information. However, there is another exosynaptic spread way, extracellular tau secretory pathway, using Golgi apparatus or not, and then vesicular, so this is a mistyping, vesicular mediated secretory pathway and tunneling nanotube. Wow, tunneling nanotube, what is that? So, and also the secreted or excreted tau is taken up by so many mechanisms here. It's been all proven by biologists and scientists. LRP1, which is also known to be involved in Parkinson's disease, and dynamine, and also pinocytosis, macro and micro. There is also micropinocytosis, and nanotube is here, and here flotillin related one, or here, and heparan sulfate proteoglycan or peptidoglycan, clathrine 1, caveolin 1. So I am familiar with that because I have studied almost 13 years nanoparticle related stuffs. So all these are used by the nanoparticles or recently viruses. And those pathogens and nano stuffs, and also body produced some the tau or paired helical filament shaped tau. So this is so complicated. Should we know that? No. But should we get exposed to this kind of information? Yes. To understand what putative, let me just do this. Putative mechanism, to understand putative mechanism, which was explained in Nature Neuroscience. Actually, this kind of information is really new, novel. And so he and his boss would have tried to understand the direct interaction, indirect via glial cells, and once extracellular, things are more complicated. Why? Because transsynaptic is a neuron-to-neuron delivery is the only one to be paid attention to. But in this case, once extracellular, there are so many players. Microglia, astrocyte, and sometimes it's a plaque. Plaque looked like a waste basket, already indolent or active. So how can you just find that? It's kind of the figure out the status of the neighboring cells. That was the one that I studied just last year. Before last year, before 2020, meaning before COVID-19 pandemic, we call it BC. Before COVID-19 pandemic, I could not save any time to understand, to follow all these, but just to kind of read only Nature, not Nature sister journal. Nature or Science, and not Science sister journal. So then we need to just pay attention to, then why are you looking for it out here? She exclaimed. So Indonesian and our Islam colleagues, the Mullah Nasruddin is quite famous. I heard, I got to know while just looking for this original contents, just like Aesop or the fairy tales or fables, and Mullah Nasruddin. Why are you looking for your purse or your key out there here? It's dark in the basement. Even though I lost it basement, it's dark. I do not look for my key in the basement. I'm looking for it in the first floor, right? Because there is more light out here. That's a sarcasm against the human behavior. Evolutionarily, we were just kind of shaped like that. So let me just show you. There is much information now. Here you can see the AD, atomic resolution structure of A beta 142 fibril, PNAS, several years ago, which is being targeted by this one, Congo red, and here AB45, fluorobetaben, and flutamethomor here, and also fluoropuronol. All these are looking at a little bit planar and not just deviated. It's just really planar. And that planar one is here, like this. So that's the information that, however, in case of the tau, there is two shapes. One is a AD form. The other is the traumatic encephalopathy one. Alzheimer fold is like this, and traumatic is like that. So meaning that a tau imaging agent is a little bit the deviated. So, but that deviation is not so much. So our radiochemists actually speculate, but did not prove, and they just kind of try to synthesize everything and then to experiment and to find out. But you know, the chronic traumatic encephalopathy has also produced pathologic tau and tau fibril paired helical filament, and then finally tangle. But interestingly, the tau imaging agents, they don't know a priori which kind of tau imaging agent is going to target AD fold and CT for that kind of issue. So we are in a kind of a trap. Our radiochemists are every day. This is a blessing for radiochemists. They need to just to kind of produce every week and every month and every year, and they found that it is not working. Chester Mattis told us that before making PIB compound, C11 PIB compound, he needed to make almost 60, or as I remember, or almost 100, and discarded, and then finally he could reach the goal to make the world first amyloid plaque imaging agent, which is useless in my opinion now. However, because the amyloid plaque imaging and tau aggregate imaging is available, we need to understand the current knowledge. We should not disregard anything which is established. So AV1 for 51, and here frontotemporal dementia, and the THK Tohoku compound, those two tau imaging agents, and also one AD imaging agents, tau imaging agents, one imaging the AD imaging agents. Here, there is a difference. So, do we change the compound, or do we use the same compound to find out the disease specificity by just the regional uptake specificity? So that's the reason why European Journal has published our colleagues, one, Samsung Medical Center and Asan Medical Center, excluding us. And they have done a marvelous job, head-to-head comparison of AV with Lawrence Berkeley people, and also Severance Hospital, AV1 for 51 and TOC compound, and they have done in the same patients. So this kind of information is showing the region and peptide specificity, peptide meaning the amyloid or tau. However, tau aggregates has been intrigued by the problem of off-target effect. And also specificity has been questioned. Not because the investigators were lazy or lenient, but because this is so complicated. No one understands correctly the problem. Only one can just summarize as a kind of a caveat now is CSF tau. CSF tau is a biomarker. However, what kind of tau is going to be measured using ELISA or chemiluminescence assay and whatever using antibodies against the CSF, the peptide molecules are not the agreed upon, meaning the total tau, sometimes total tau amount is less than the phosphorylated tau. Why? Because tau is used to be truncated, and terminal is lost. The total tau meaning the N to C terminal, so its amount is underestimated. However, phosphorylated tau is just attacking only the phosphorylated portion. So that's the issue of the biomarker. You know, we are the kind of best person to understand that. Why? Because we are experts in radioimmunoassay and immunoradiometric assay. We meaning the first generation. The next generation leaders in the Asian countries, you belong to the second generation in Asia, and you might not be quite familiar with the biomarker. However, simpler is better for patients. So if the proteomic analysis, how expensive it is, I mean the whatever expenses it incur. So if it is going to be just kind of a blood sample or saliva, then it might be better for patients. That's the idea of the clinicians. Okay. Now let's move on. Then let me explain to you by just the searching with a short time on Saturday, just 5 days ago. On Saturday, I started to, this question popped up in my mind. Off-target effect. I asked this to Professor Yun-Sang Lee, and he did not answer clearly. So I just looked through the literature, and I could find cross-interaction of tau PET tracers with monoamine oxidase B, evidence from the in silico modeling in vivo imaging. That and then silico modeling is based upon simulation, free energy binding, affinity, and cross-reactivity something. And they have used the sulfonamide, and whose structure is fitting into the here. And then they have used the upside down image in another figure. So I just rotated a little bit and turned it. So the letters are in different phase, but the interior is the same. And here, THK, Tohoku compound, and PBB3, and Roche compound, and T Siemens compound, and Merck compound, those kinds of Johnson & Johnson compound, all those things are well captured. However, their kind of location is a little bit different. Dihydroepinephrine or something. So this is for monoamine. That's epinephrine is monoamine, and monoamine oxidase substrate. So we have already C11DED, previously HED, which was routinely used by Technical University of Munich, meaning TUM. Munich, the group under the leadership of Professor Schweiger in German, Schweiger, and the Marcus Schweiger. They have they popularized this one, and also THK 5317 binding here. That compound is here. Because the monoamine oxidase enzyme is quite prevalent in here, deep gray consisting of the striatum, thalamus, striatum meaning the putamen and globus pallidus, And of course, cowled nucleus. Okay, so those kinds of information had been shown by this paper, but this paper has clarified the difficulty of producing the basal ganglia would have been suspected to be an off-target. Why? We all know that the sensory motor nucleus, sensory motor area of the frontal and parietal lobes, and also occipital lobe, and also basal ganglia are spared, and cerebellum and pons are spared. So in FDG PET imaging, so if some agent is targeting better the basal ganglia, they would have known, radiochemist, even the radiochemist or radiopharmacist would have known, even though they do not know, they would have a very good clinician colleague, meaning the nuclear medicine physicians, we know. So that, so we can say that that kind of, some kind of cross-reactivity is suspected. And then they try to find out by just comparing the structure of the compound they have developed. That might have happened, but there is no the report. So this is only my speculation. So, however, modeling is modeling. And all these should be verified. It takes time and it takes money and it takes labor. Sometimes, as I told you, NIH Eckermann has spent his last years in a kind of not so happy mood. Why? Because cholinergic hypothesis has been just kind of turned into a newly developing one, which is amyloid hypothesis. And then tau people challenged it. So 25 years ago, when I see the science news and briefs, and there has been an explanation, there is a hot dispute, amyloid beta group and also tau group. So it's for a kind of a diversion or joke, they have told it A beta meaning Baptist, Christian, and tau people is Taoist. The rhyme, according to the rhyme, it reminds us that the Taoism, the traditional Chinese religion or the belief structure, like Confucianism, something. Okay. So now I'd like to ask 최은별. 최은별 있어요? 켜요? At first, I'm going to ask in Korean, and if she or he is answering in Korean, then I would just kind of translate. So you can just. So now I think I have defeated. I have make the opinion by Dr. Gill obsolete, obliterated, obscured. So I'm going to ask you. So before going into the students, and 최은별 안 왔나 봐. 답이 없네요. 신진섭. 그래서 AD의 다음 번째 타겟은 뭐라고 생각해요? 여기 써놨죠. 뭐예요? 자, 골라 잡아 하나. 이게 뭐 남대문 시장도 아니고 막 골라 그냥. 아밀로이드하고 타우 놓고 고르더니 골치 아프니까 저기서 잃어버렸지만 여기 가로등 밑에서 찾는다. 그러는 이솝우화 비슷한 우화를 설명한 다음에 뭐라도 고르라고 지금 그러고 있는 거예요. 이거 이렇게 우리가 공부해야 돼요, 이렇게? 아니, 과학자들이 다 하면 그때부터 우리가 하면 안 돼? 그렇게 했더니 내 평생이 다 가고 말았는데, 나 21년 동안 브레인 했는데, 우리나라에서 타우시피어하고 그 타우에 대한 아밀로이드에 대한 치료제였나? 그치. 모노클 앤티바디하고 그 타우시피어까지 도입해가지고 했어요. 그리고 이것 때문에 페데마라이도 도입했어요. 참, 뭐야 근데 지금 싸우고 있는 꼴로 봐서는 학자들은 지금 비판하면 좋아요. 아니 자기들이 말되는 약을 만들어낸 것도 아니고 뭐 약 돼가지고서는 FDA에서 온갖 비난을 무릅쓰고서 그래도 바이오젠은 미국 회사잖냐. 우리 한번 해보자. 아무것도 안 하고 있기를 아리셉트만 먹이고 있기에는 좀 너무하지 않냐. 이렇게 많은 사람이 점점점 다 돌아가고 있는데 일종의 말은 compassionate use예요. 다른 방법이 없으니까 그거라도 해보자 하는데 근데 왜 9년 동안 그것만 하냐. 거기다가 돈을 엄청나게 쓰겠다 그러니까 돈 문제로 이제 보일 수도 있는데 반대하는 학자들은 쉽죠. 그렇게 하면 안 된다 그러고 반대하는데 그럼 대안 있냐 그러면 없는데요. 이렇게 되는 거거든. 자, 그래서 신진석 골라봐요. So let's make a kind of a census. So I'm going to say to you that microglia is the next target, or astrocyte, or infiltrating immune cells are the next target. So let me just tell you that there is an information. Infiltrating immune cells had been just calmed down by GlaxoSmithKline. I forgot the company. Pfizer, or there is a drug against multiple sclerosis. Multiple sclerosis is a disease of the infiltrating immune cells, especially B cells, via a CSF, not a systemic one. So that's based upon the recent information they published in Science and Nature, which I read frequently. And then based on that information, then infiltrating immune cells might be a target. So even though there has been many reports proving the infiltrating immune cells are doing harm to the Alzheimer's disease, the development and progression. Okay, so let me just do with the students. 신진섭 제일 마지막에 대답하고요. 이소희. 자, 그래서 마이크로글리아 아스트로사이트 인필트레이트 이뮨 셀 어느 개 찍을래요? 이소희. I'm just kind of inviting every student by their name. 이소희.
네, 교수님.
네. 이 중에 어느 거예요?
네?
이것 중에 어느 게 알츠하이머 디지즈 치료하는 타겟이냐고, 표적이냐고.
저는 마이크로글라이아라고 생각합니다.
오케이, 됐어요. 그다음에 김혜원 뭐예요?
저도 마이크로글리아라고
오케이.
생각했습니다.
김수빈 뭐예요? 김수빈.
네, 교수님.
네, 뭐예요? 셋 중에? 김수빈이 답했어요. 강제당했다고 셋 중에 하나 고르라는 법 없죠? 네? 우리 여론조사할 때도 그렇게 안 해요. 내가 김수빈이 5번째 답을 말했기 때문에 여섯 번째 답을 말했기 때문에 4번하고 5번을 가르쳐 줍니다. 4번 all of the above, 5번 none of the above, 6번 모르겠다. 7번 microglia and astrocyte, 8번 astrocyte and infiltrating immune cell, 9번 마이크로글리아 앤드 인필트레이팅 이뮤셀. 그래서 김수빈 학생이 본의 아니게 그 나머지 선다 초이스가 있다는 것을 밝혔어요. 이게 얼마나 놀라운 일이에요. 아밀로이드하고 타우하고 고르라고 그랬을 때 그렇게 얘기하지 말라는 법 없었거든요. 그렇게 연구하는 사람들이 있었어요. 아밀로이드 엔지오파티 등등 해가지고. 자, 됐고. 김수빈은 그래서 6번이다 이거지. 모른다. 임경현. 임경현. 학생한테 꼭 말을 시켜야 되겠어요. 지금 1시 46분이지만. 이거 원래 코스웍이에요. 임경현, 안 왔나 보구나. 저기 그 불출석 이렇게 최유리 교수가 체크하고 계시죠. 서현주부터 신진석까지는 아까 다 온 걸로 체크됐으니까. 서현주. 뭐 같아요? 서현주가 없구나. 영어로 쭉 하다 보니까는 에이 모르겠다 그러고 다 나가버렸나 봐. 그래서 김동빈 씨 녹화 기록은 잘 해놓잖아요. 유튜브에다 안 올리는 겁니다. 이거. 김동빈 씨도 자리에 없나 보다. 조혜란 씨 안 올리는 겁니다. 전해줘요. 여기 이제 앞으로 나올 데이터는 우리만 갖고 있는 거가 많아서 아직 발표 안 한 거라서 못 올려요. 자, 그러면 서현주, 황지은.
네, 교수님.
뭐 같아요, 이거?
저는 셋 다 고르겠습니다. 네.
강민준. 뭐 같아요? 자, 이제 알겠죠? 30년 동안 저하고 3학년, 의과대학 3학년 학생들이 실습할 때 왜 일부 학생은 좋아했지만 일부 학생은 극도로 싫어했는지. 여러분이 절대로 어디서도 볼 수 없었던 문제를 물어보고 답을 요구하는데 미리 알고 있을 거라고 생각 안 해요. 3학년 오면은 뭐 어떻게 알겠어. 환자를 본 적 있나, 뭐 검사를 본 적이 있나, 그러니까 모르는데 상식적으로 생각해 봐서 뭐 같으냐. 그리고 그때 변은 뭐였냐면 의사는 모든 환자를 다 보고 나서 의사 노릇하는 게 아니거든요. 환자는 굉장히 다양한 방식으로 자신의 불편함을 호소하거나 보여줘. 그러니까 전혀 상관없는 것 같은 데서 그 실마리를 찾아내서 도울 일을 생각해내야 되거든. 그러니까 3학년 학생이 임상 시작했는데 답하지 말라는 법이 있냐고요. 그리고 답해보면 맞는 답이 나오는데 그 당시에는 아니래도 좀 있다가. 그래서 어디까지 갔지? 황지은 답했죠? 안 했나?
했습니다, 방금.
네.
네.
예, 셋 다라 그랬지. 강민준 뭐예요? 강민준 있는 거야, 없는 거야 도대체? 11명만 데리고서 한 학기를 했으면은 참 좋았겠죠, 그렇죠? 최유리 교수가 반쯤 가르쳐주고 내가 반.
여기 근처에 원정들 학생 대상으로 해서
강민준.
다 옮길 수 있는
네, 이거 보세요. 이렇게 해서 그냥 패시브하게 듣게 해놨더니 라디오 듣는 거랑 똑같아. TV도 아니고 라디오입니다. 라디오 듣는 거랑 똑같아서요. 라디오는 아니고 보이는 라디오를 듣는 거구나. 왜냐하면 화면은 보고 있으니까. 그렇게 똑같아요. 그래서 이거 학습 효과 없습니다. 제가 코비드-19 이후에 줌으로 예과 의학을 위한 수학 응용을 2학기를 했는데 세 번째 학기는 지원자가 없어서 없애버렸고요. 이번 학기 두 번째 학기는 두 학기를 했는데 학생이
The professor is gonna be research.
학생이 10명이어야 되고요. 계속 발표를 해야 돼. 그래야 학습 효과가 무지하게 높아요. 근데 이게 4, 50명 되면은 절대로 장악 안 됩니다. 그리고 그거는 존속이 안 됩니다. 아무리 좋은 컨텐츠를 해보세요. 뭐 전 세계에 얼마나 많은 컨텐츠가 있는데 유튜브에 다 올라가 있지. 자, 신진섭 파이널.
네, 교수님.
뭡니까?
저는 처음에 인필트레이팅 이뮨셀이라고 생각했는데요.
네.
이제 보기가 많이 늘어나다 보니까 all of them이 정답일 것 같다고 생각을 바꾸기로 했습니다.
네, 지금까지 아주 말을 잘했는데 맨 마지막에 정답이라고 얘기한 거가 틀렸어요. 세상에 정답이 있으면 물어봤겠어요, 이거. 그냥 가르쳐주고 말지. 네? 그러니까 희한하게도 김수빈 학생이 모른다가 두 가지 뜻이 있거든. 알려져 있는지 모르는데 나는 모른다. 인데 미안하지만 알려져 있는 것도 없고 아는 것도 없어요. 그런데 우리는 알아내야 되거든. 그래서 그나마 지금 신진섭 학생의 다른 점은 뭐냐? 최리 교수하고 지금 4학기째, 3학기째인가? 배우고 있으니까 어떻게 되냐 하면 틀리더라도 자신 있게 틀려. 그게 우리가 일단 도달해야 되는 겁니다. 그 의견이 생긴 거죠. So I just exercised a question and answer with my 10 students. 2 or 3 ran away. Every time in every class there is a runaway. That's okay. So which is going to be going to pay by the kind of Professor Cheuri, another professor who is residing here. And then among those 5 to 6 students, 1 or 2 students said it's all of them. So we need to understand the microglia and astrocyte infiltrating immune cells, all of them, and to test which is a beta target, because all of them are targets. Some suspected microglia. Why? Because even though they do not know very much, because this would be their first year graded course or the second year graded course, they think they understand the astrocyte and oligodendrocytes. 아까 9번 말고 10번이 있었는데 빼먹었구나. 여기 있는 거 아무것도 아니고 10번째는 뭐냐면 다른 거다. 그래서 엔도텔리얼 셀이다 하는 그룹이 있어요. 그게 아밀로이드 엔지오파티야. 그게 아밀로이드 엔지오파티입니다. 오케이, 그래서 저 이제 여러 가지 의견이 several the 2 or 3 said that it might be microglia. Why? Because they are innate immune cells. Then do we understand microglia and astrocyte infiltrating immune cells very much, or even stromal cells? No. Then had been there any good methods to understand the microglia and others? No. Then previously, but recently, is there any kind of light on the horizon and light along a long tunnel? Yes, that's kind of a spatial something, spatial one, which is no. And interestingly, it was kind of like this. The microglia was classified as a homeostatic microglia and disease-associated microglia. And this was a arrogant naming. Why? Because here they imply that disease meaning the Alzheimer's disease or the neurodegenerative diseases, but brain afflicting diseases are so many. So the scientists or investigators in other disciplines were so mad, upset by this one. And anyway, the name was so implying, meaning them. DAM, people immediately think that DAMN. Okay, so, and then astrocytes, they try to name another disease-associated astrocyte, but it was too late, and reactive astrocytes. And somebody had published a paper in Cell, and then all the others gathered together. No, Nature Neuroscience, and the next year, all the others gathered together. No, disease-associated astrocyte is a too much bold, too much arrogant naming. So we need to call it reactive astrocytes. And infiltrating immune cells, the problem is really there. Meaning, and this one. And if those cells are going to be the culprit of harming the brain, in Alzheimer's disease, is at least in animal model, then it is enough to understand the role of the microglia or others. No, we need to find which surface target is going to be, and then which kinds of the method are going to be working against the surface target. For example, microglia is well known to have a CSF1 receptor. CSF1 receptor is not cerebral spinal fluid. It's colony stimulating factor 1, and it has a receptor, and CSF1 has a kind of a ligand activity and if we block CSF1 receptor, then all those microglia are down, and then only once they are rejuvenated. If the second dose and then microglia cannot recover, at least in animal model. Then recently, CSF1 receptor is also acting against is targeted by the interleukin 34. So people found that the interleukin 34 is in the area of cerebellum, and then CSF1 is all the other areas. And recently, again, the CSF receptor 2, CSF2, was also found. CSR2 was also found, and CSFR2 was found to be the syndecan, already characterized receptor in other molecular biology area. So that's kind of complicated. It's too complex for the outsiders like our students. Their the expertise is not going to be neuroscience. They are just kind of trying to understand a little bit of neuroscience so that they can just do their own discipline very well, like applied bioengineering or the molecular medicine and biopharmaceutical sciences. And however, most of us belong to the nuclear medicine. So we are interested in and doing the imaging. So for imaging, we need also surface target. So how can we define the surface target and which kind of cells? That story have a crooked, long and crooked, and the pitiful, shameful history here, TSPO. So TSPO is well said to be the reactive microglia. Really? So recently, there have been a development, and the fancy, marvelous development. And interestingly, last year was the COVID-19 year. And in another sense, in another community and in science, in the field of science, spatially resolved transcriptomics. Until spatial had not been put to the transcriptomics was not our the focus of attention. However, it's become it has become the spatial transcriptomics, and it is going to yield functional neuroanatomy imaging on microscopic slides. So our interest has started. So the young professor Hong Yun Choi, who is not here, he adopted that discipline, as I remember, December 2019. Now we have obtained, we meaning my group, Professor Cheuri and me, our group has acquired almost 30 the mouse animals which were sacrificed for science purpose, for the sake of the science progress. So that transcriptome for exploration of targets could be summarized with this one slide. This one slide is same, and the last slide is Nature October issue. Last month, I have received the Nature. They try to find out the difference between human and animal in the structure of the cortex. You might have known the cortex in animal, they call it isocortex. In humans, it's called neocortex. Anyway, cortex just the upper side of the corpus callosum. It's or corona radiata. It's called neocortex. They try to find the transcriptome can be a good tool to predict morphological difference, physiological difference, and that's their conclusion. Strong correspondence between this one. However, for each gene and surface or intracellular protein, they have already reported 10 or more years ago, messenger RNA abundance and protein abundance is just like this. Grossly, they have what? Yes, they have a correlation. However, with the same amount of messenger RNA, it's a log scale. Protein abundance is so different. So actually, this kind of correlation does not mean anything per individual gene and its product. However, we don't have protein or proteomics tool for 100,000 isoforms of protein of each cell. But we have now tool for 22,000 in case of human or mouse, 1,000 mRNA measuring capability like this. No, this one, transcriptomics. This was a patch sequencing, which is a specialized form, but we call it spatial transcriptomics. And then that spatial transcriptomics has been reiterated or recapitulated by another paper after 7 years. Predicted protein is closely correlated. So if there is no other better method, then why not use the transcriptomics to understand? So we've tried that. Professor Che and Che, two professors gathered together. They have published recently hippocampal glucose uptake here, FDG uptake here, is not due to brain cells, I mean the neurons or neuron astrocyte pairs. It's because of microglia. And also the similar paper, microglia activity, FTG PET, the correlation, including autoradiographic study, was published by the München University group, not technical university, and Science Translational Medicine. They were more lucky to publish their paper. Why? Because their paper is more basic. So in addition to this information, they have used this transgenic mouse and so many the difficult to achieve, even in South Korea, they have done. So that's microglial contribution. So based on this one, in Alzheimer's disease, so while looking at that kind of information, among those students, microglia student would think that, I have one as a kind of a question and answer session. Really? So let me show you. In Scientist, that's a web-based journal, a Indian woman scientist based on Bangalore, she wrote down here, microglia was first identified around this age, this year, 2000 microglia, because of their clear close relationship to the macrophage, classified this one. This has been, this paradigm broke down in the face of such variation. So at least what you are saying, microglia is so many. However, target TREM2, CSF1 receptor, and SIGLEC, and then rest in peace, RIP. No, that's a joke. And interleukin-1 beta and progranulin on LRRK. These kind of things are in the phase 2. So if we do continue to do only amyloid plaque and tau imaging, so aren't we Mullah Nasruddin? We have tools. We do that one after 20 years. That was not the case. It happened. It happened. The Eckermann's case. Muscarinic receptor imaging is going to be the kind of holy grail and final solution. Why? Because there has been developed the Aricept, which was approved by what? US FDA. Now, still now, there are so many the market, so huge market. So was it Novartis or Merck and Pfizer? And they are getting wealthier and wealthier. And people are in frustrating situation. There is no way to avoid that drug. Why? Because there is no drug. So so there was the case. So should we develop any the TREM2 or CSF1 receptor and SIGLEC and that kind of the ligand? So you know that, but as you know, the development of a drug, radiopharmaceutical against anything, is really a kind of a lotto type of job. So we need a real good motive and also the insight and luck. she said, single-cell and spatial transcriptomics will allow researchers to narrow down on the genetic signature of microglia that are involved in disease. Why? Inherent differences are so many, and if microglia is the sole target, differences in activation state during disease, which depend upon specific pathologies and the location. So the taking care of or taming those microglia who are harmful to make the disease worse was looked so difficult. So should we just depend on our radiopharmacy experts or the company radiopharmaceutical, very small number of radiopharmaceutical companies, or the Hans Wilgen Bester type of, I'm just saying the FAPI inventor, FAPI ligand PET inventor, the Hans Wilgen Bester type of expert. You need to take care of your 166 million people in Bangladesh, 260 million people in Indonesia, 100 million people in Vietnam. Should we depend upon the big pharma company, multinational pharma company, or scientists? No. Either giving up our hope or leading them to the right direction. So if your country has more people, then your responsibility upon your shoulder are heavier. In this country, we have 52 million, including North Korea, 72 million. Okay. Then what about the astrocytes? Astrocytes are really kind of an innocent bystander? No. Interestingly, they have found recently in recent report in Cell, one of the best journal, the neurons and astrocytes are very close. They do exchange glutamate and glutamine, and even they do exchange a toxic lipid. That's the reason why ApoE isotype, ApoE the ApoE4, 3, 2 are the best known the kind of predictor of Alzheimer's disease in humans. So until recent, they have also found that the lipid is a problem, and amyloid is also the heading 4 here. Where is it? Amyloid was heading for, the glutamate receptor 2. So amyloid, if A-beta are there, so many in extracellular space, then it is going to excite neurons, recipient neurons, and so that's the reason why seizure susceptibility is coming higher, A-beta dependent hyperactivity, and tau is trying to just calm down, but they fail finally. And so in this neuron and astrocyte model co-culture system, they could find lipid was just kind of made and transferred to the astrocyte, and astrocyte, the mitochondria is really big, and then they just kind of take care of the excess lipid. That's the reason why astrocytes are best known the producer of ApoE, apolipoprotein E. However, apolipoprotein E is produced any kind of cells in the brain. So that's our first experimental research. Here, in upper Nature Review Immunology, several years ago, early state disease, this kind of situation is there. It's kind of imaginary drawing based on, and middle state disease, successfully, that's contained. A beta is harmful, but that inert amyloid plaque is not harmful. But by looking at only the morphology of the amyloid plaque, there is no way to find out. And then nearby neurons are going to be damaged. So, here, you are the first person outside our lab to see the real distribution of our ApoE messenger RNA here in 12 animals. 2 animals were cut in coronal and 4 animals in wild type with various the drug treatment, and please disregard that. Animals were cut in side-style way. And here you can see the hippocampus and thalamus here, hypothalamus here, striatum here, and isocortex there, and also amygdala here, and whatever. So the same. And then the lower row was another the mRNA, another genes and mRNA, but the ApoE was so profuse. So that ApoE is important and lipid is toxic. So if you find during your PhD work, if you find this one, wow, I'm going to study this, and then report to the scientific community that ApoE is the culprit, then some of them would respond, we already know that. And some of them would respond, no, yours are doing one. Mine, I have another, the option, like our students. They have, when they were pushed, when they were kind of encouraged to pick one, just so they try to find microglia. And then finally they found that, there might be a collaboration between those cells. Then why not all of the above? Then I just included another option, none of the above, or 2 of them, 3 of them, or 2 kind of pairs of any kind of combination. So do we know that? So scientists in this field knows everything, and we are the only person who do not know that? No. Unfortunately and fortunately, meaning fortunately and unfortunately, it is not the case. So we need to understand, and spatial transcriptomics is just at the beginning. We are the best person to read this kind of ambiguous, unclear image. Yeah? And clear image is our best policy. Actually, the radiologist would have difficulty in reading this kind of image. But we are the one. We are functionally oriented, and even though transcriptomics are not quite concordant, it's just crudely, coarsely correlated with the peptide, we are happy. Why? Because we have been using FDG for almost 3 decades, and then we don't know FDG is taken up by reactive microglia or neurons, or even astrocytes, or even oligodendrocytes. We had been happy to say, here FDG uptake, SUV max or FCV average or something, and then we can exactly say that this area is the posterior cingulate area, and this area, if there is a decreased FDG uptake, then this is kind of an ominous sign of the initial phase of the Alzheimer's disease. And posterior cingulate, we don't know why. And then amyloid is propagating or not. People did not give any evidence that amyloid is propagated. But the tau is propagated either way, exosynaptic way and transsynaptic way, from here to there. But interestingly, Braak staging is saying both the amyloid and the tau. So that's the reason people are confused. Usually nuclear medicine physicians or neurologists, they are confused to say that the amyloid is propagated. No. In Parkinson's disease at first, and at first in prion disease, and then tau. At least I know that, but I'm not saying that amyloid is not. There has been a proof of amyloid is not propagated. That's absence of proof, meaning no proof of. Actually, it's not proof of no presence. It's no proof of, it's after almost 1.5 hours, I'm feeling a little bit hypoglycemic. Then I'm going to just ask, as I just so now you are at the top? No. Where is it? Let me just, here. I promised to ask Alexander, can you turn on your, unmute your microphone? Yeah. Yeah, okay. Yeah, so let me just tell you, iodine 125 Clinda is one of the drug aiming at the TSPO. And wild type is like here, green, and then the light blue, and transgenic AD model mouse here, astrocyte, and microglia like this, and 12-month and 24-month endothelial cell like this. Then by looking at this one, in this image, TSPO imaging, so here, increased uptake is here. You see, then that uptake is because of the first, the choice of answer is astrocyte. Second choice is microglia. Third choice is non-astrocyte, non-microglia. So which one is your answer? It's just kind of looking at the bar graph. It does not need any the astrocyte or microglia physiology or TSPO one. So it's just kind of 2020 Journal of Alzheimer's Disease.
Maybe microglia.
Microglia. So I did not say this is from 24 months, the transitioning mouse. You immediately found that there is a kind of break there, almost to the infinite amount, and microglia is there. Okay, it's correct. Then, what about the astrocytes? Is there any possibility this activity is from the astrocytes?
Yeah, actually, yeah.
Yeah, of course. So it's because here they have they have put their title, astrocytic TSPO upregulation appears before microglial TSPO. Yeah, microglia has a basic level of TSPO, and astrocyte increased. So by just looking at the increase, however, using this TSPO imaging of translocator protein upregulation is selective for pro-inflammatory polarized astrocyte and microglia in the Journal of Glia. So if somebody is writing down the TSPO was originally developed for this model here, 24-hour, excessively increased the TSPO in microglia. They have made it, but when they were used for imaging, interestingly, and we need to think also about the propensity of astrocyte and microglia. This is a in vitro study. So in vitro study does not say anything about the in vivo one. 2 points. One is in vivo situation, numbers of astrocytes are high, numbers of microglia is low. Then there is a kind of gradient, I mean the difference. So even though this small amount, because astrocyte, this kind of increase, if, yeah, you know, what I'm saying is just that's single astrocyte, single microglia, or 10,000 cells in the culture flask, culture well. But here, astrocyte is 10 to 1, astrocyte and microglia. Actually, microglia in the homeostatic state, they don't have anything to do. It's just kind of it's kind not there is no need to run homicide detectives if the safety is good on the street. There is a kind of only the traffic police. Okay, in 10 to 1, then this might be astrocyte. This kind of a spatial and in vivo thing has not been declared in case of the single cell RNA. Now it has become spatial. And then transcriptome data is well correlated with morphology. So if you're going to use TSPO for targeting microglia, it should be here, right? Only 24 more months. But, you know, when do we need to use the neuroinflammation imaging? It's MCI or very early stage to use at least NSAID drugs. Non-steroidal anti-inflammatory drug is well known to just kind of prevent, and in some the mysterious way. So this would be the last slide of the second session. Interestingly, using transcriptomic data only, segmentation was clearly similarly done in all the animals, 30 animals, and I have shown to you only 2 animals each. Even with that, we could find there has been some kind of a section blunder. I mean, the frozen section is blind cutting. So here, interestingly, in humans, I would say it's a Sylvian fissure. In mice, there is no Sylvian fissure. So we still suspect what might be. Anyway, layered the organization of the isocortex. However, interestingly, TSPO was not so high. It's really scattered. You have seen the second time, you have already seen here, apolipoprotein is so densely expressed, but like this, but here. So this can be best studied by the imaging specialist. Finally, I think we need to just transfer our knowledge to pathologist, meaning the tissue pathologist, not clinical pathologist or the laboratory medicine expert. Laboratory medicine expert, they are the clumsy in reading images. Their images are, in case of the immunofluorescence, it's just grade 1, 2, 3, 4. That's all. But the tissue pathologists, they are recently doing similar things as ours. And why we are interested in this kind of job, we need to select a correct target. TSP, if we have seen TSPO long ago, their transcriptomic pattern, then I don't think I would choose this one as a target. And if we just have experiment like this, and if we just have done like this, here TSPO is upregulated in both specifically pro-inflammatory polarized, then we don't know what we are just kind of challenging. Then there is no way to say that this might be the drug target for disease-modifying drug. So that kind of radiopharmaceutical is not going to survive. Cross-reacting, but interestingly, fluorodeoxyglucose, FDG had survived. They do image M1 macrophage, granulocyte. They do image the tumor cells, and they don't image the well-differentiated hepatocellular carcinoma cells. Interestingly, they are more for the poorly differentiated one. So that's the reason why our colleague in Hong Kong, I forgot his name. Yeah, so he just invented the use of the C11 acetate to differentiate well-differentiated hepatoma from poorly differentiated FDG-RB. So that's the end of the second one. And then I have gone through 19 slides here. So I'm sorry. I think Karishima and Andreas Lim and so the other people might have been the involved, might have participated as a kind of second and third and fourth, and including Dr. Asmar did. But interestingly, here, let me just find it. Here, there is a kind of some kind of breakage. So, so I don't need to just go on. So you might have understood. So we now know I have been the almost a designated reviewer of neuroinflammation articles invited by the Ignacy Cario of European Journal back in 20 years ago, around 2005 or 2010, Ignacy Cario sent all those French articles to me. So I was bored by the TSPO imaging. And as it turned out to be interesting, after 20 years, I'm just kind of accumulating evidence against the TSPO. TSPO is not a good. And TSPO is the mitochondrial outer membrane the protein, and which means that the chemical should go through the cellular basement membrane. At first, a blood-brain barrier, and then ISF interstitial space, and then ISF interstitial fluid space, and then meet the cells, and then cross the cellular basement membrane, and again mitochondria outer membrane. Oh, so it's better than the intranuclear the localization, because they need to the nuclear nuclear membrane. Nuclear membrane is a special one, has a special nuclear pore complex. Now, so sorry for the letting wait, but according to my original plan, this might be the kind of inflection point of this talk. And then I think I have a successful experiment with the excellent next generation leaders of the one. So some of the transcriptomic images are still under development and not published. So removing those images, I'm going to, previously I did not plan to just upload any PowerPoint slide except for the previous blank one, like this blank one. But I'm going to just, why? Because I think, including my students, you have followed me well. So you would have produced your own the bias or behavior against or for the current progress and situation of the AD theranostics or therapy development and their imaging. And especially you might have the flavor of understanding of TSPO imaging. And TSPO was the first one to be analyzed seriously, because I know Clinda, I know the PK 11195, 94, 95. So, and others, meaning G compound and whatever. That the G compound is General Electric. So and then let me just tell you one more. So you're going to finish this one. Here we have 224 historical participants. So if you allow us, then we will send your kind of hub leader people for this communication, and also they will send the announcement to all of the emails. So if you allow me, and if you don't want to receive any more, then just to kind of send back your email. Please remove. Why? Because you have information in a duplicated fashion. But what I'm saying is, among 200 people, maybe 180 people, 80 next generation leaders would be more involved in tumor studies. That's the current situation. Only 10% or less would be for brain studies, even though it's amazing and interesting. And if it is not going to be translated to the clinical situation, then it's useless. If we are paying our more attention to the science and the knowledge and curiosity, then we would not have chosen the medicine. We have chosen medicine to help people. So if it is not going to be a help, then we are so cold, so sarcastic to remove any kind of diversion from science. Science is going to be okay. So let me finish here. And then next week, in addition to the current one, I have made this kind of cerebrovascular disease Q1, Q2, and cerebrovascular disease Fact 1, and cerebrovascular disease Q3 is an addendum to the epilepsy Q1, pitch radiopharmaceutical, epilepsy Q2, imaging method, and then professors have kindly made a Parkinson disease summary question, and also amyloid tau imaging summary and biomarker, and his own for the tumor or correlated imaging related one. So this is going to be much easier for nuclear medicine, the experts, and then we are going to do exercise. I'm not going to expand the slide number. Why? Because it is already 26 to cover next week. So the person, and then today's paper, the list of the today's the participants are going to be kept again. In addition to 200 or more, then I will I will exercise the pronunciation of everyone. So my goal next week is to invite everyone just to once or more, except for students. Students today, all the students were just invited, and they would have enjoyed. Just kind of guesswork. The science is a guesswork. Science is not a sequential job. It's kind of a copy. And then finally, when we make a draft or manuscript, we just make it scientific way. Okay, so our students would have experienced that one. Okay, today's the 51 site, the excluding the students, and almost to 35 or 6 site has been connected. And then I just ask Professor Hall. So finally, if I'd ask if there is any comment or question. Yeah, Faresa Pradana and the Deborah Get and Megawati May Kantoro, also Para and Pinka, and then those people. And you don't need to be worried about. The question might be today's one. So there is no prior knowledge, but you can just kind of read. So, or I have put the references. So as a kind of luxurious diversion, you just read on the internet. So then participate, then you'd be a little bit more comfortable with what I'm saying. Okay, if there is, let me just put up chatting. Okay, nothing. So please, Professor So, at this time point, we are not going to just continue the questionnaire. Yeah, so we can resume that if the Asia Connect project, European Commission colleagues, European Union colleagues would allow us to do that one. Right now, the announcement is not right here. So questionnaire, please don't deliver any load to the participants. Participants might just kind of listen and then imagine and then raise a hand and kind of a census and then just forget, right? And then it should be a little bit more fun than the kind of a duty or responsibility. Okay, there is no question, and then I would just conclude as is promised before 2:50. See you next week.